Good morning. For those of you who are joining very promptly, we will be starting in another minute or two. Thank you. Morning, good afternoon, and good evening. I am very pleased today to introduce our ICAP grand rounds on long-acting injectable cabotegravir for HIV prevention, promises and pitfalls. And I will take a moment to tell you about our speaker, Dr. Raphael Landovitz. Dr. Landovitz is a renowned infectious disease specialist and clinical researcher. He's a professor of medicine at the University of California, Los Angeles's David Geffen School of Medicine, and director of the UCLA Center for Clinical AIDS Research and Education. Dr. Landovitz has focused on the optimization of the use of antiretroviral medications for both HIV treatment and prevention. He's been part of the leadership of the United States government's National Institutes of Health and the NIH's AIDS clinical trials research networks. And this includes serving on the executive committee of the HIV Prevention Trials Network, the HPTN. He leads several biomedical and behavioral prevention intervention studies and projects assessing post-exposure prophylaxis, PEP, and pre-exposure prophylaxis, PrEP, strategies for men who have sex with men and cis and transgender women. He led the NIH-funded phase 2 and phase 3 registrational clinical trials evaluating long-acting injectable cabotegravir for PrEP, and these studies contributed to the recent approval by the U.S. Food and Drug Administration. He continues to study other investigational long-acting and extended-release products for HIV prevention, and I think you'll all agree this makes Rafi really just a perfectly positioned person to talk to us about long-acting injectable cabotegravir for HIV prevention. Just a quick reminder to everyone to put your questions in the Q&A box, and we will be sure to take a little time at the end to answer questions. Rafi.
Thank you so much, Dr. Justman, and thank you to you and Dr. Frank and Dr. Al-Sadr for inviting me, and good morning, good afternoon, good evening to everyone. It's really an honor to be here with you, and I look forward to sharing some thoughts and insights about long-acting injectable PrEP as we think about how to transition from clinical trials to implementation. If it's okay with everyone, I'm going to stop my video now and share my slides to optimize everyone's bandwidth here, and hopefully everyone will be able to see the slides. If Dr. Justman, are you able to see the slides? Okay, great.
Perfect.
Okay, thanks so much. So these are my disclosures, and I know this group probably does not need to be reminded of this, but just to set the context for this discussion, of course, 40 years on into the HIV pandemic, despite remarkable scientific discoveries for both HIV treatment and prevention that has reduced the morbidity and mortality associated with HIV infection tremendously and given us tremendous advances in prevention, we still have an unacceptable number of new HIV infections every year globally, mostly concentrated in Eastern and Southern Africa, Eastern Europe and Central Asia, and Latin America. As we think about how we're doing with pre-exposure prophylaxis uptake 10 years after the first definitive evidence and regulatory approvals for oral tenofovir-based PrEP, you can see from this map that we failed to meet UN targets for number of users by 2020, and we really have not done an equitable job of getting PrEP to all parts of the globe for all populations. So we clearly have a lot to do to optimize and maximize the prevention efficacy of this extremely powerful intervention. And I think we still struggle with understanding the behavioral and social science correlates of use of both existing and some of these newer products that we're going to be talking about. What's particularly frustrating about those inequities and our lack of being able to meet those global goals is really robust evidence at a population level that if pre-exposure prophylaxis is made available at low or no cost and with minimal or no barriers to access, you can have extraordinarily impressive levels of population level reductions in HIV incidence. And this has been demonstrated in cities and regions across the world with really impressive results. And I'm sure many of you have contributed to these data sets that led to these observations. And it's really something to be celebrated and really I see as a call to action for all of us to make sure that safe and effective PrEP interventions are made equitably and widely available without barriers to populations across the world. That's the only way that we're going to see these kinds of reductions in incidence and potentially meet our goals. Just to remind everyone, I think many of you who have seen or heard me speak before on the topic of pre-exposure prophylaxis know I love this graphic. When I put up tables of clinical trial results demonstrating the effectiveness or efficacy of oral PrEP, I find it very overwhelming. So I've created this construct where the sex at birth of the population at risk is shown by the figure in each of these trial boxes, and how filled up the figure is the point estimate of the reduction in HIV incidence of that particular trial. And you can see that with oral PrEP at the beginning, there was a wide variety of clinical trial results that led to a lot of confusion and consternation on the part of both providers and ministries of health, ethics committees, and consumers of PrEP about whether this intervention, an oral tablet with antiviral activity, could actually be effective for both rectal and vaginal routes of exposure. And of course, we learned that the devil's always in the details. And in this case, the details, of course, that matters is if you don't take an intervention as prescribed, it's not reasonable to expect it to work. And we have an enormous amount of work to do to fully understand the reasons that people can't, won't, or just don't take oral PrEP medications as prescribed. And in the last 10 years, we've developed a relatively sophisticated understanding of some of the reasons why this is particularly challenging among some populations. We've also learned that a pericoital on-demand or really behaviorally congruent way of administering TDF-FTC, at least for MSM and trans women in the ICAP study, was also a highly effective method of delivering oral PrEP. And fascinatingly, some guidelines are now really challenging us to expand and extrapolate the data from ICAP to apply its recommendation for use to all men. So I think this is a fascinating advance that we can talk about in the questions if people are interested or have thoughts about that. Of course, also in the US, we also now have an approval for TAF FTC, tenofovir alafenamide FTC, for cisgender men and transgender women. And really, the US regulatory approval is for all routes of exposure except for vaginal. And we're hoping for data that will support vaginal exposure protection in the not too distant future. It was in that context, really, of this observation that currently available PrEP regimens could have the potential for really tremendous population level reductions in HIV incidence, but we had uneven and inequitable uptake of these interventions, that the search really began for additional items on the menu, if you will, for pre-exposure prophylaxis. And of course, some of these challenges with adhering to a daily oral tablet really prompted the focus on long-acting interventions, either delivery mechanisms or products that wouldn't require a user to do something on a daily basis. And it was in that spirit that a number of long-acting injectable medications went through early development. The one, of course, that moved the furthest and that we're going to be focusing on most today is cabotegravir, which is a novel integrase inhibitor. It's chemically similar in structure to dolutegravir that most are familiar with as part of HIV treatment regimens. And this integrase inhibitor is available both as a daily oral tablet and as an injection for gluteal intramuscular injection. And I'm saying that quite specifically because at this point, we really only have data for its use when injected in the gluteal muscle. And that's suboptimal for a number of reasons. And we can come back to that again in the chat and the Q&A at the end. But obviously people who either have implants or fillers in the gluteal region can't safely use a gluteal injection in this way, and also it makes it very challenging to self-administer, and a number of other circumstances make it prohibitive. So we have a ways to go because of that limitation. But this was studied in two large efficacy studies, HPTN 083 and 084. The difference between the studies was mostly population. 083 was done in cisgender men and transgender women who have sex with men. And it was done in the countries you can see listed on the bottom of this slide. And 084 was done in cisgender women in the global south. Again, the countries you can see listed here. But they both had a very similar design. It was an active controlled trial, so no one got a placebo intervention, which is really an important point because everyone was at risk for HIV acquisition by risk criteria for entering the study, but it was double-blinded. So everyone got both an active intervention and an inactive intervention. So people either got active cabotegravir or they got active daily oral TDF FTC, and they got placebo or dummy interventions for the other one that they weren't randomized to. There was a 5-week oral lead-in period, and then every 8-week injections for about 3 years. And then we quote unquote covered the pharmacokinetic tail as people stopped the injections with a year of daily oral TDF FTC, as shown in this slide. Hopefully everybody's at least heard about these trial results. On the left is 083 and on the right is 084. And I'll be brief in summarizing this because I'm sure many of you have heard these results. The top line, both from the blinded period, both these studies were stopped early by an independent data safety monitoring board for high levels of efficacy of both arms of the studies. In 083, there was a 66% reduction in HIV incidence in individuals randomized to cabotegravir compared to daily oral TDF FTC. And in 084, this really staggering result, a more than 90% reduction in HIV incidence in the cabotegravir randomized women compared to the women randomized to TDF, FTC. And I point that out as particularly staggering and remarkable just because we have struggled so hard in clinical trials to see these levels of product usage that could accomplish these very high levels of reduction in HIV incidence among cisgender women in the global South. And so this really is a nod not only to the product, but to the concept of things that are culturally and locally and contextually appropriate, where injectable interventions are really looked at as one of the most effective interventions. So when I talk, I'm going to now pivot a little bit and talk about what some of the surprises and challenges have been with long-acting PrEP and cabotegravir in specific. This is a little bit of a complicated slide. So what I just want to remind everyone, and I'm going to focus for a second on when PrEP that's long-acting fails, when it doesn't work, when there are breakthrough infections, what we discovered is it's harder to detect using what I'm going to term conventional HIV testing algorithms. That means the usual tests that we do to diagnose HIV infection. And so let me orient you quickly on this slide. On the left side is what we did in HPTN 083 and 084 at the sites. And so when people screened for the study, everybody got a point of care rapid test that had to be negative. They got a laboratory-based, so a blood test for antigen antibody, and that had to be negative. And they also got a viral load, an RNA test, and that had to be negative before they entered in the study. And that RNA had to be within 14 days. When somebody enrolled and then at follow-up visits, everybody had at least one rapid test and a laboratory-based antigen antibody test. If any of those tests were positive or it was equivocal, the sites did have the ability to do some additional tests through the study, including viral load and a DNA test that we had done centrally at Johns Hopkins University, but was resulted to sites to help disentangle confusing results. But the RNA testing, the viral load testing, was not part of the routine initial algorithm. When it became clear that someone had broken through PrEP in one of these studies, that is acquired HIV, we had stored samples from every visit, and we went back at the HPTN laboratory center, and we did a really complicated and extensive set of tests that were all done on the same platforms to make it uniform across all of the studies, and that included qualitative RNA testing and quantitative RNA testing. And then we tested backwards from that first time that the RNA testing was positive until it was found to be negative definitively, in order to establish the first moment that we had evidence of breakthrough of the PrEP regimen. And then we had an independent blinded committee of people not involved in the studies who helped us adjudicate this really big panel of results. So let me show you a couple of examples of what we saw in HPTN 083. And again, I apologize, this is a little complicated, so let me take a minute and just walk you through it. I promise there's a reason I'm taking you through this detail, because this really has implications for programs and how we are going to be rolling cabotegravir out. Because doing a lot of these sophisticated and expensive tests isn't feasible in a lot of the locations where cabotegravir is going to be used. So what I really want to do by showing this to you is taking a peek under the hood of what we found and understanding what could be going on when we can't do some of these complicated and sophisticated tests in programs. So each of the pictures that I'm going to show you coming up has this same format. So this graph that's on the bottom of the slide is going to tell you weeks since enrollment in the study. And the vertical axis, the y-axis, is the cabotegravir concentration in plasma. And the orange circles that go along tell you those markers of the cabotegravir concentrations at each site, at each time point. And if the dots are not connected, like you'll see in that first period on the left side of the slide, that's because that was when someone was taking the oral lead-in to establish the safety and tolerability of CAB before getting any injections. And then when the dots are connected, that was after people started injections. Each injection is a vertical sort of neon green line. And when the testing at the site turned positive is the blue line. And when using this very extensive centralized retrospective laboratory testing found the infection to have first occurred, that's the red line. So I hope that makes sense. And on the top that's overlaying it all are the details of the centralized testing. So you can see what the viral load was, what the antigen antibody test was, what the Genius confirmatory test was at each of those time points. And the reference point that the black arrow on the right side of your slide is pointing to, that's what we call 8x PAIC 90, which is about double what the non-human primates, so the animal models of protection, indicate we want the concentrations to be for protection in this sort of setting. So here's an example of somebody who sneaked into the study despite actually having HIV at enrollment. We did our best using RNA testing to make sure that people didn't have HIV when they came into HPTN 083, but of course nothing is perfect and people can have ongoing exposures and you can miss it. And here's an example of one that was missed. And what you can see here is the person, look at the orange circles, took their oral drug quite well. You get high levels when we measured them at the 2 oral time points here that I'm sort of using my cursor to show you. And then they got their first injection. And then in the first follow-up visit after that first injection, the site found a positive test. And so they stopped the cabotegravir appropriately. And when we went back on the retrospective samples, it became clear that there was evidence of infection all the way back, even before they started any PrEP medication. So this wasn't a PrEP failure. They had HIV when they entered the study. And what's sort of fascinating is the viral load was 50,000 at enrollment, but the antigen antibody test still didn't catch it. And I think we think of these antigen antibody tests as being able to catch these viral loads when they're that high, but they didn't in this case, and we don't fully understand why. The other thing that you can see here is cabotegravir essentially as monotherapy for treatment of that HIV infection that was undetected was really powerful, at least in the short term, at reducing the viral load from 50,000 down to 204. Why the site-based testing and the antigen antibody testing turned positive here at almost week 9 with only a viral load of 1,600, but it didn't pick up at 50,000, we don't know the answer. But what I want to point out here is by the time we caught it at the site using the conventional testing, these resistance mutations indicate integrase resistance. So this would be expected to compromise integrase inhibitors as per a first-line treatment. So that's one example I want to show you. Now that you have the schema familiar, I'm going to go a little bit quicker. The next example I'm going to show you is somebody who acquired HIV during the pharmacokinetic tail. So they took their oral drug, they did fine. They got their injections 4 times and they did fine. And here, then they didn't have any injections for about 3/4 of a year, 217 days. And when they came back into care, they were found to have broken through PrEP. So they got infected sometime during this long, slow decay period of the cabotegravir. And when we detected that infection, there were no delays. There was nothing funny about the diagnostics. The test caught it with a viral load of somewhere between 50 and 93,000. And it was wild type. There was no integrase resistance. So we've talked a lot and we've worried a lot about people who might get HIV infection during the tail phase of cabotegravir, but that at least in this case, and at least in all of the cases we've seen so far, we didn't see that. This person was able to be suppressed on an efavirenz-based regimen. The next thing I want to show you is sort of the one situation in which you can get resistance in the context of a tail phase acquisition. And it's not actually from acquisition during the tail. So again, here's someone, they took oral drug, they got a bunch of injections, then they disappeared for over a year. They came back, and the first set of testing, when they were back, they were actually in the window period, unbeknownst to anybody. So it wasn't caught, and they got 2 more injections before the site testing returned positive. So when you have someone who's in the window phase and you restart cabotegravir, you absolutely can get integrase resistance. Now, happily, this person did not have resistance to other classes. So again, they were able to be suppressed on an efavirenz-based regimen. Those of you who are wondering, how do we know that this integrase mutation didn't occur from acquisition during the tail? It's because the very first genotype that we have from the samples right when the HIV was detected, they were actually wild type. The integrase resistance came up after that. So we do know for sure that this was sort of caused by the inadvertent administration of 2 more injections after that initial restarting of the injections. Many of you know that the oral lead-in has been made optional in regulatory approvals. And in fact, in the ongoing open label extension of the 083 and 084 studies, and I'll tell you more about that in a second, we are making it optional as well to get safety and acceptability data of making that oral lead-in optional. I'm actually someone who's a strong believer that it's probably in most cases preferred to not use the oral lead-in, and here's why I say that. What we know about people who struggle with oral dosing is that people who struggle with oral dosing probably aren't going to do well with a month of oral pills. So in this particular case, this is somebody who acquired HIV during the oral lead-in. What you can see is at the 2 time points we measured cabotegravir during the oral lead-in, they seem to have okay levels. But remember, we don't know what happened between these levels. Someone might have had breaks in adherence. We just don't know what happened there. This person didn't have any detection of their HIV and actually went on to get 2 injections. And finally, when we detected the infection, you can see, again, we had integrase inhibitor resistance already. But when we back tested, you can see that the infection was actually there before they ever got their first injection. But our conventional diagnostic algorithms did not catch it. So again, this is somebody who we might be concerned would not have responded as well to integrase-based first-line treatment. So this is one of the cases, and I'm only going to show you 2 more, I promise, where somebody got HIV infection despite perfectly on-time cabotegravir injections. We now have 6 examples of this throughout the 083 trial, which I know that sounds like a lot, and a lot of us are very sort of fixated on trying to understand why these cases failed. But the most important thing that I can assure you of at this point as we interrogate it further is this is a very rare occurrence. This is occurring at an incidence rate overall in the trial of 0.17%, this breakthrough despite on-7 injections. It's an incredibly rare event. So I know we focus a lot on it and we muse a lot about it because we don't understand it fully, but I really can't emphasize that enough, that this is a rare event. But this is somebody who took their oral drug just fine. They got injections just fine. We detected the infection here at about week 41 of the study. But when we went back, it looks like it was there since about week 27 on the study, but with very low levels of RNA, 6.1 copies, and then it went to negative. And really, this person, it was only very late as the cabotegravir washed out that we were able to see this infection using regular diagnostics. And again, integrase inhibitor resistance was found at the first genotype that we were able to do when the viral loads started to come up even a little bit. This person actually didn't, because these diagnostics are so hard to interpret, they didn't believe that they actually had acquired HIV. And they actually went on a 4-week course of PEP in the middle of all this, which may have made it even harder to diagnose the infection. So this is going to be a challenge to all of us in rolling out these long-acting PrEP agents. So it's something to think about. The last one I want to show you is something that is very interesting. This is someone who had at least one 10-week delay between their injections. Now, a 10-week delay potentially compromising efficacy has not been something that's proven yet. I can't promise you that that's what led to this infection. And if it does end up being 10 or more weeks after which efficacy is lost after an injection, that is likely to not be the case for cisgender women, where the pharmacokinetics are very different. Remember, this is from 083. But this is somebody who was somewhat erratic in getting their injections. And interestingly, with these erratic injections, there was no delay in the diagnosis. The testing all turned positive at the same time, and it was wild type. And because it was wild type, you can see that they were able to suppress on an efavirenz-based regimen also. So when I sum up sort of what happens with resistance to these integrase inhibitors versus not, it really depends on what the clinical context is of when the breakthrough infection occurred. And so what we've sort of done is sort of summarize here what we know about it. And let me sort of walk you through it very quickly, and then we'll move on to some implementation concepts. If you start cabotegravir, so the A cases, A1 through 4, with undiagnosed HIV infection, or you restart it with undiagnosed HIV infection, basically there's about a 1 in 3 chance of there being integrase resistance in that context. So that's a pretty high rate. If you break through during the oral lead-in period, 2/3 of those cases we found integrase resistance. If you break through with on-time injections, all of them in our hands had integrase-based mutations. If you broke through with erratic injections, nobody had resistance. And if you broke through during the tail phase, at least 6 months from the last injection, nobody had resistance. So I hope that's helpful in just summarizing what we know so far. We're going to get a lot more information now with clinical use and rollout, but this is sort of at least how I've codified my understanding of when you are likely to get integrase resistance versus not with cabotegravir. Now, all this testing observations and the RNA stuff has led to the US FDA and the US CDC to recommend that these RNA tests be part of HIV testing algorithms for the use of cabotegravir PrEP at every injection, both before you're starting and in monitoring. And moreover, they specifically say that you should use an RNA test that is FDA approved for HIV diagnostics, which really limits you only to 2 tests. Now, I do want to say that the WHO, the Australian regulatory approval, the Zimbabwean and the Ugandan regulatory approvals do not have this requirement. They all indicate that just the standard of care HIV testing that is part of programs in each country is to be used at the time of initiation of cabotegravir PrEP and prior to each injection. So we are going to get a lot of more information about how quickly we can detect these infections without using RNA testing, as was done in these studies and is being required in the United States with these programmatic rollouts. So it's really important that we have careful surveillance to understand how that works and what the implications are for resistance and then subsequent response to first-line HIV treatment, because we don't know the answer to that. In our studies, we don't have anybody who was really treated with an integrase inhibitor-based regimen after failing cabotegravir PrEP. So these are the US clinical guidelines about what tests are done where, and we're doing STI testing every 4 months instead of every 3 months, as we are with oral PrEP, depending on the population. That's for highest risk populations. The US recommendations say every 6 months for heterosexually active cisgender women and cisgender men. But it's changed our clinic flows significantly to try and implement this changed schedule when we're used to administering pre-exposure prophylaxis at quarterly visits. This is more contacts with the healthcare system in order to administer these injections and do this follow-up. So, you know, in the US, a lot of people ask me, when we have multiple items on the menu, how do we make good decisions without good data? Some people have said to me, because both 083 and 084 had a superiority result, does that mean we have a moral or ethical obligation to put everyone on injectable cabotegravir? And what's the best PrEP? And my answer to that is the best PrEP is whatever the patient will adhere to and persist with the best. And that's acceptability, that's preference, that's context, that's stigma, that's cost, that's access. It's so many things wrapped into one, and we'll come to that in a second. But there is no moral or ethical obligation to use one product over the other. And I really want to be clear about that. People ask me with cabotegravir, what's the time to protection? And I want to be very clear, we don't know. But pharmacokinetic analyses, and Vive presented this late last year at one of the PK conferences, is it seems like the time to what we think are protective concentrations, the median, is about 2 days and 95% by 7 days. So, you know, I don't know the answer for sure, but it's likely somewhere in that range. It's also likely different slightly if you did the oral lead-in versus not. You may be slightly quicker with the oral lead-in because you have residual drug if you go to the injections right away. And how long does it last? I'm not sure. You can see I made some suggestion that maybe it's 10 weeks for males. We don't know. It's probably 12 or more for females. And if you do have breakthrough on injectable, long-acting injectable PrEP, I've summarized for you what we know about resistance. If it was a recently acquired infection, so an injection in the last 6 to 12 months, I would tend to recommend using a boosted PI or an NNRTI. But can you use an integration inhibitor-based regimen if it's longer than that? I believe the WHO is recommending that TLD still be the first-line treatment regimen for people who are going to break through on cabotegravir PrEP. So we're going to get a lot more data about that strategy coming up. So right now, the 083 and 084 studies are in open label extension. So they're ongoing. We're getting more data, and we look forward to sharing those with you as soon as we get them. They made the oral lead-in optional. We are using RNA screening at all visits to see if that's a good thing or a bad thing. And we will report back as soon as we have more information. But I really want to end with a couple of slides, really with a call to action here. And I need to thank Mitchell Warren from AVAC, who has really, he and the AVAC group have really been leading us in thinking, even while these trials were being conducted and before they resulted, in how do we learn from oral PrEP rollout and try to do better. We really need to be aggressive with looking at the post-approval implementation studies. We need to learn from what we learned from the oral PrEP rollout. We need to make sure that the studies that are being designed now for cabotegravir and other long-acting agents are designed to address the questions that policy and decision makers need to make. We need to make sure that they're done expeditiously to inform decision making at global and country levels, and make sure that the complex and fragmented stakeholder landscape really are engaged at all levels of these discussions. So where do we need to go, right? We need to make sure that these decision maker questions that are coming up at the country level are addressing these next set of studies as we move towards implementation. We need to make sure that these are being planned even as we are still gathering data from these clinical trials. And it needs to be really a shared strategy with diverse stakeholders, or this will not be successful. And we're destined to repeat the same mistakes that we made with oral PrEP. This is a really complicated and busy slide that I'm not going to go through in detail because I want to make sure to leave time for questions and discussion. But this is sort of a summary, and we're happy to share these slides at the end of the presentation. I know the ICAP team will be sharing them. Again, this is another Mitchell-Warren AVAC slide that really goes through some of the priorities that the group has identified, and a call to action for what we need to do to make sure that this is integrated into PrEP and prevention programming at country levels with appropriate and a full stakeholder engagement and research that is ongoing to inform the questions that implementers and decision makers need to appropriately make those decisions. And, you know, the AVAC group really, I think, has this beautiful schema of breaking down the needs and the conflicts where providers and health systems and what they want and what users want. And I can't emphasize enough that choices of prevention products are going to remain critical to the success of our prevention efforts, even as these products go through their clinical trials and implementation phases. If we don't have a menu of products, we're not going to do well. There need to be coalitions designated in each body that is representing these decision makers, accelerating access. There have been example groups convened by UNITAID, the WHO, UNAIDS, Global Fund, and PEPFAR. AVAC is assisting, and they're really hoping to be able to coordinate key stakeholder activities to prevent the mistakes with oral, or delays, rather, let me say delays more than mistakes, of oral PrEP implementation as we learn more about this product. So for each product, we need to understand the clinical correlates of its use, what it implies for policy and programs, and what it means for people, because it's never going to just be the product, it's the whole program, and just having an efficacious product isn't going to solve everything. The funders need to know a lot very quickly, including cost and cost effectiveness. Programs need to know what policies need to be put in place in order to deliver a new product, and users need to create demand and have a lot of questions about how the product use will be rolled out, what will it mean for them, and what will the consequences or benefits of product use be. All of this needs to be addressed if we want to prevent these delays that we've seen with oral PrEP. So what now for Cabotegravir PrEP? I mentioned that it's approved in the US, in Australia, in Uganda, and Zimbabwe. There are regulatory approvals pending in a number of other countries, with even more to come, which is all very exciting. But I stole this slide mercilessly from Al Liu at UCSF, who loves to remind us quite rightly that unless we make analogy to family planning and analogy to what is good and what is challenging and what is not so good about a variety of interventions, we're not going to get broad uptake. And we need user-friendly mechanisms like this Bedsider website is for family planning to give people the information they need so they can make informed choices about what prevention product they want to use. And there really is rigorous data in the family planning field that the more options you have, the more use you will get. And we believe that this will also be true for HIV prevention. So the last challenge to us is that we want to translate these really exciting clinical trial results and observations into real viable choices for users, providers, and health systems. We have to understand the optimal testing strategy and initiation needs for these long-acting PrEP agents. We really need robust civil society engagement as we go to implementation, research, and program planning. We really need procurement and commodity funding for launch and ongoingly. provider training, participant and consumer demand development, realistic targets for these interventions, not just coverage, and understanding what these new products can fix and what they still can't fix, and what we need to make the research agenda for ongoing prevention programs and products. And finally, the last thing I promise I will say is that we need to make sure we do better, more equitably with this introduction, with the injectable and also with the ring, than we did with oral PrEP and with COVID vaccines. I'd like to acknowledge the HPTN 083 sponsorship, the teams, our pharmaceutical partners, these really critical implementation discussions that have helped frame this, the HPTN leadership, our team, site staff, and of course, our participants, families, and communities. And thank you all so much for your attention. Happy to answer questions or take comments. Thanks so much.
Well, Rafi, that was really just terrific. Thank you very much. So we do have some questions that have come in, although I will just start us off and ask you a little bit more about the number of, I'm going to call them breakthrough infections, that you walked us through the different scenarios, but I wondered if you kind of can take a step back. What was the total number of breakthrough infections compared to the total number of individuals in the trials, and perhaps even if there's a way of comparing it to the total number of infections that were averted? Again, all of this is in the RCT setting, which I understand is different from sort of a more real world setting.
Yeah, thanks, Jessica. So I'm gonna take the liberty of defining true breakthrough infections as people who broke through on-time injections, right? So the total number is 6 in 083. And that is out of, so of the cabotegravir treated participants, we had 4,566 total. So half of that, so 2,200 some odd. We did an incidence calculation on that to help try and get a better idea of how frequently this occurs. And that's 0.17 per 100 person years. We haven't done infections averted calculations yet, but we did do a number needed to prevent or number needed to treat, and it's somewhere in the 120 range. And that's in comparison to number needed to administer to prevent one infection with cabotegravir compared to TDF FTC. That's not compared to nothing. I hope that's helpful. That's as granular as we've gotten as digging into that yet.
Okay. I mean, I think, you know, we can probably all agree that these are, while they're concerning, they're very small numbers. And I think we, you know, want to be clear that the message is that the cabotegravir in a randomized control trial setting was incredibly effective.
Correct.
People should not feel really afraid to use it, despite these small numbers of breakthrough and so on.
Correct. And I would remind everyone also that for cisgender women in the global South, it was even rarer. They have not seen any breakthroughs among cisgender women in the global South with on-time injections.
Okay. Now, one of the questions that did come in was asking about whether it's possible that any of these breakthrough infections were caused by resistant strains. So, yeah. Yeah,
it's a great question. And in the majority of those cases, we have been able to do sensitive research-based single genome amplification with colleagues at University of Pittsburgh. And in the majority of the cases, we're able to demonstrate wild type virus before the resistance emerged. So we don't believe that is certainly not the majority of the cases. There are one or two that we weren't able to amplify and get a genotype before we found resistance, or it was just there at the first viremic sample. So those I can't assure you that it wasn't a transmitted integrase resistant variant, but global surveillance suggests that that's a very rare event to date, which is great news. So while I can't rule it out, I'm a little dubious that that's what accounted for this.
Okay. There's a question about the effect of cabotegravir on feminizing hormones and vice versa. Any information on that?
Also a great question. So the short answer is we don't know bidirectionally for sure. We know that gender-affirming hormone therapy does not affect gender-affirming hormone. We know that gender-affirming hormone therapy does not affect cabotegravir concentrations. Beatrice Grinstein presented those data at the Montreal AIDS 2022 conference this past summer. The question of whether or not cabotegravir affects gender-affirming hormone therapy concentrations has not been definitively answered. We do know that cabotegravir does not affect hormonal contraceptive levels. I'm fully aware that that's not the same thing. And studies are being designed right now to specifically answer that question in a rigorous way. We can't answer it in 083 because we did not provide a uniform and carefully measured set of gender-affirming hormone therapies to measure it. So it's a whole myriad of different regimens that.
Okay. Just seeing some additional questions that have come in. Let me ask you, I think you alluded to this at the earlier portion of your talk about the location of the injections in the gluteal muscle. Are there studies going on to look at alternate dosing sites, such as the thigh or the arm? Where do you think that's going? You're on mute, Rafi. You're on mute.
Oh, thank you. Sorry. I want to acknowledge that that's an absolutely critical issue. It is being looked at in the thigh. Already, I believe data are collected and complete, and we hope to see them soon for people with HIV using cabotegravir and rilpivirine as treatment. One of the ongoing registrational treatment studies did have an addition that looked at thigh injections, and they're comparing the pharmacokinetic safety and tolerability to the gluteal injections. And again, we hope to see those data very soon. I believe, and I don't want to speak out of school here, that a phase 1 study in the prevention space, so people who do not have HIV, is also either about to begin or has just begun to look at thigh injections as well for pharmacokinetics. The challenge is it's a very large volume injection. By very large, I mean 3 milliliters. So you need a large muscle belly to inject it into. So there probably is not going to be an arm option, but the thigh, we hope, will be an option. And I think for me, the big question is, are the pharmacokinetics the same, and is it as tolerable? Because, you know, the safety concern that really the only safety concern that came out of the cabotegravir PrEP trials were injection site reactions. And, you know, these happened in a lot of people, the majority of people, but they were intolerable to a very small number. but there were some people who just found them very painful.
Yep. We have so many interesting questions. We're not going to be able to get to all of them. Let me try to combine a couple of them into sort of the general direction of implementation at a wider scale, both the challenges in terms of the type of diagnostic testing and the very frequent measures of HIV viral load that's required, and also thinking about task shifting and who will, be able to give these injections and who will be able to interpret all of these frequent diagnostic tests. I recognize it's a whole talk unto itself, but any brief thoughts in that direction would be really appreciated.
Yeah, no, thank you for that. I think it's a really complicated question that requires a lot of people way smarter than me to think about. But let me break that into two realms. The question about differentiated service delivery. I think, we have a lot to learn from family planning clinics who've been doing this for a long time. I realize that in the majority of contexts, the HIV prevention and treatment services are currently not integrated into those clinics, but I think we can learn from how those are set up, what we are likely going to need to have in terms of staffing to administer and follow people with these injectable regimens. The diagnostic test issue is a huge quagmire, and I think the 30,000-foot answer is we need advocacy and an enormous call to action for more sensitive, inexpensive point-of-care diagnostics. And some of you have heard me say this before, but if we can create a COVID vaccine and at-home COVID tests in, 12 to 18 months, why can't we, get better point-of-care, inexpensive HIV testing done on a similar time frame? I'm not sure what the answer is to that, but until we get that, I think we have to do the best with current in-country algorithms. I want to remind people that the site-based testing in 083 and 084, which were rapid and a blood-based antigen antibody test, demonstrated, right, was the engine that generated this incredible superiority result in both trials. There's an incredible amount of nuance here underlying it with elegant laboratory work, but the bottom line is you still got this incredible superiority result just using conventional testing. I think we just need to be very careful in surveying for what the implications are going to be for subsequent response to first-line treatment. And I think there needs to be a high level of suspicion if people particularly have been delayed in getting their injections. But if somebody's been getting on-time injections, these failures are quite rare. And so we are going to be struggling with, you do enough tests in a low pretest probability context, you're going to have false positives. And, that's going to cause a lot of discussion and challenges and anxiety for both patients and providers, I fear.
So actually, I may have sort of misrepresented things. The frequent diagnostic testing is at this point part of CDC guidelines in the US. Where has the WHO come out on this?
So the WHO has recommended a conventional algorithm before starting and at the time of each injection. So it still is more frequent, but it's just not a different set of tests than we are currently using to implement oral tenofovir-based PrEP.
And in our very last minute here, anything that you can share with us in the experience with the open label extension trials so far?
Yeah, thanks. We're still very much in the throes of it. The one thing I can share is when we made the oral lead-in optional, in the open label extension, I was anticipating that no one would want it. And I was wrong. What we're seeing, and these are early data, so this is not, by any means a final result. So I would hope everyone would take it in the spirit of just sort of information sharing. About a third of people are choosing to use the oral lead-in. And I was surprised by that. And I'll be very interested to see, as we delve into this more, how much of that is driven by the patients or participants themselves, and how much of that is driven by their providers. Because remember, there's multiple layers there in that decision making.
Great. Okay, well, I think we are right at the top of the hour, and so we will close, and thank you for really a wonderful talk and a great round of questions. The webinar recording and slides will be posted on the ICAP website. And I'm going to just provide a reminder for our upcoming World AIDS Day event, where we'll be talking about the latest insights into the global HIV response and the importance of leaving no one behind. That will be Thursday, December 1st at 11 a.m. Eastern time. There'll be both in-person and online opportunities to join. So thank you, Rafi, and thank you to our ICAP communications team for organizing all of this, and look forward to seeing all of you on December 1st. Take care.